What part of the gene?
Kind of, like the siRNA blocks gene expression. Yeah, while the microRNA blocks translation.
All right, are you ready to get started? There was a lot of information yesterday, and so I wanted you to start by going back and thinking about some of this to try and see what you remember. So just a quick review. A control element is a part of the DNA. It's upstream of the promoter part of a gene. So upstream means kind of backwards from where transcription is going to happen. And it's where other proteins, it's where activators and repressors, bummer, can attach. Activators and repressors are proteins that either speed up and help with transcription, that's an activator, or slow down or turn off transcription, that would be a repressor. So activators and repressors are proteins. They attach to the DNA at the control elements, and then they either help the other proteins and RNA polymerase bond at the promoter part, or they make it harder for that to happen. So the transcription either happens more efficiently or less efficiently. The bottom two are two of those types of short RNA molecules that we talked about that can also be part of regulating how genes are expressed. The exact mechanism of how they work is slightly different. mRNAs, that stands for microRNAs, bond directly to the messenger RNA and literally block the transfer RNAs from bonding, so they don't allow translation to start. The cRNAs, that's also what we looked at in that video yesterday about RNAi. That's another name for the same thing. They called it the COP in that video. Those are small interfering RNAs. They also prevent the beginning of translation, but they actually they bond to a messenger RNA and mark it to be broken down and degraded, so it will never be translated. Like they said, it kind of just destroys all of the copies of that gene within the cell. Yeah?
Do you know what variation of siRNA or RNAi that you can test for these?
Most likely you'll see this form.
Okay.
They probably won't say RNAi. I put it in your learning targets and notes because that's what that video refers to it as, but probably you'll see this because this, I'm pretty sure, is what's in your textbook.
Okay.
Yep. Other questions? So the next question to kind of lead us into today's topic. Activators and repressors, we said, are proteins that bond to the control elements. mRNAs and cRNAs, of course, are short RNA molecules. Turn and talk to the person next to you quickly. Where do all of those molecules, those activators, repressors, and those different types of short RNAs, where do they come from in your cell? Or how does your cell make them? Turn and talk about that. Think about that. I know.
Because they would get the instructions and guide how work and send it out on the chefs where the guide.
Well, isn't the question asking where did the chefs come
Where
Okay. I feel like I just stretched your brain a little bit with that question. Where do these come from? Yeah, it all goes back to DNA. You were probably thinking too hard. This isn't a crazy complicated question. What I wanted you to realize is that even the things that control transcription of other genes are also somewhere coded for in the DNA. Activators and repressors are proteins. That means somewhere in the DNA, there's a gene that tells that particular cell to make that particular activator through transcription and translation, and that activator then goes to a different part of the DNA and attaches on there and turns on another gene. Those small RNAs are made through the process of transcription. There's a code in the DNA that has the sequence for those small interfering RNA molecules, and it makes those on purpose to go out and block translation of other proteins as a way to control the amount of another protein in the cell. So there's lots and lots of levels. One of the things you'll realize about biology is you can always go back another step and say, well, where did that come from? Well, where did that come from? And we're kind of going one step further back today.
So how would the gene be turned on to make an activator with activators?
Some genes may be able to, like some transcription happens even without extra activation. Like not all genes need to be turned on. Some are just automatically transcribed, and that's part of what we're going to talk about today, why certain parts of the DNA are transcribed more than others. And sometimes these activators and repressors don't completely turn it on or off. They just speed it up or slow it down to make more or less of it. And one of the other factors, so part of the point of having you think about this is to realize that we can keep going back and to realize that all of this is always way more complicated than it seems, even though it seems plenty complicated. But today we're going to talk about one other way that cells know what parts of the DNA to read and use. Some of it has to do with the combination of activators and repressors that are found within that cell. But those also need to be made by the DNA. So there's more to it than just that. Some of it has to do with how the DNA is packaged in the first place. And we kind of mentioned this and alluded to this previously. We talked about the fact that the double helix, big long strand of DNA has to wrap around these histone proteins in order to fit in the nucleus of your cell. If I remember right, every single cell in your body has something like 2 meters of DNA in it, which is like the height of you in one cell. And so obviously it has to be wound up pretty tight to fit inside of the nucleus of your cell. So it wraps around these histone proteins that clump in these bundles called nucleosomes. It's several of those histone proteins all together with DNA wrapped around them. Then when the DNA has to tighten up more for cell division, those kind of coil up on each other tighter and tighter and tighter into a chromosome. But the part that we're looking at today is this section. This is chromatin. And so that's kind of how DNA exists when the cell is in interphase most of the cell's life. The DNA is coiled around histones, but it's not super, super, super tightly wound up. That only happens during mitosis. And you can see if you look at that part of the DNA in that square, some parts of the actual molecule of DNA are easier to get to than others, right? If you think of all those other molecules, all those transcription factors and the activators and the repressors and the RNA polymerase that all need to get into that DNA and bond to it to start transcription, it's going to be really hard for all of those molecules to get to DNA that's really tightly wound around those nucleosomes. Those genes that are found in these areas of DNA don't get transcribed very much. The parts that are the most accessible and the easiest to transcribe are these stretches that are wide open and accessible. And the way that the DNA wraps up, specifically which sections are wrapped tight or left open, is different in different types of cells because of chemical tags that are attached to those histone proteins. So today we're actually talking about a layer of regulation that goes beyond just the sequence of bases in the DNA. Today we're talking about epigenetics, which is something you might have heard a little bit about in 10th grade. We're just going to talk a little more about it now. This is one of those areas that scientists have only recently started to kind of figure out, that it's not just the code of bases that you have. There are other factors beyond that that can affect how your traits are expressed and what you inherit. So epigenetic inheritance has to do with chemical tags that are attached to the histone proteins that either loosen up the DNA to make it easier to transcribe or tighten it back up so that it's harder for it to be read. So the two main types of modifications that affect this process are the addition of acetyl groups. An acetyl group is COCH3. You don't need to know that, but that's what an acetyl group is. It's just a chemical group. And so histone acetylation means this chemical group is being tagged onto those histone proteins. That in this picture is probably these tags right here, because when those are added, it tends to loosen up and open up the DNA so that transcription is easier. The other type of modification is the addition of methyl groups, which are probably the other ones in this picture. These tags that tend to wrap DNA much more tightly so that it's harder for transcription to happen. A good analogy that I heard for how to think about epigenetics, because epigenetics, these chemical tags that are added on, don't actually change your DNA at all, right? They're not changing the sequence of bases, but they're changing your cell's ability to read your DNA. And so the analogy that I heard for this that I really like is if you imagine like a book you're reading or a textbook, the words on the page are like the code of the DNA, right? What epigenetic markers are is like if you take a highlighter and highlight the important parts, and you take some whiteout and you white out the parts you don't need to read, those are epigenetic marks. The text is still there. It's all still there underneath. You've just kind of highlighted the parts that are important and covered up the parts that aren't. That's what epigenetics does. Questions about that basic idea? So epigenetics is a really new field of research that's being very actively explored, and people are learning tons of stuff about this. These chemical tags can be changed and modified by all kinds of different factors that have to do with your lifestyle, the chemicals you're exposed to, your diet, the amount of sleep you get, the amount of stress you experience, and these can change throughout your lifetime. We're going to watch a TED Talk today that talks about epigenetics, and we'll give you some examples, some what I think are pretty cool examples of some studies that they've done with epigenetics, and kind of how they're looking at this and some of the reasons that they think this part of how genes are regulated can be so important, okay? This TED Talk is almost 20 minutes. I think it's like 18 minutes. And it's the last official part of the lesson today. So after that, we'll be finished a little bit early, and you guys will have some time to work on mastering biology that's due tonight or review for tomorrow's assessment. I'll put this up right now just in case people at home go home and do their video at home. These are the things coming up to work on mastering biology tonight. There is an assessment tomorrow. This might be the last one that we do through the College Board website. I'm still figuring out exactly what we're going to do next week to wrap things up, but I think it's going to be a little different. You also have your mitosis lab that's due Friday. So after we watch this video, you'll have some time to work on those. Folks at home, just like yesterday, I will throw the link into the chat here, and then you are welcome to sign off and watch this video on your own. And then I will start it for all of you.